for those of you who don't know me, my name is Jean-Pierre Desforges. As I was introduced, I'm currently doing a postdoc at McGill, but I'm happy to announce my new position as an assistant professor at the University of Winnipeg in the Department of Environmental Studies and Sciences. And for you who aren't familiar with my work, I've essentially been working with marine mammals, specifically in the Arctic, for over a decade now, looking at questions of contaminant effects and increasingly trying to figure out the impacts of multiple stressors. But for today, I'll be specifically talking about some of the research that I've been fortunate enough to take part of over the past couple of years, looking at mercury and climate change and how they interact to affect marine mammal populations. Here we go. So specifically, I'll just start by talking about how Arctic ecosystems are undergoing rapid environmental change and what that means for stressors like contaminants, mercury, and kind of how that interacts with climate change. Then I'm going to go through 3 studies that I've been working on really over the past 2 years only. The first one is going to be looking at narwhals and how we can use their tusk to look back in time. The second one will be looking at beluga, a very similar study but using teeth. And then the third one is going to be focusing more on the impacts of mercury rather than looking at temporal patterns. And we'll be looking at the effects of mercury on the brain of several marine mammals. And then I'll just kind of finish off by talking about what we've learned from these studies and kind of what's left to know or what we don't know. So I likely don't have to convince this audience that humans are having widespread impacts on ecosystems around the world. Now, we all know there's various ways that humans are changing the natural world. Some of them are summarized in these figures to include things like climate change, chemical pollution, invasive species, habitat destruction, etc. But importantly, we know kind of increasingly that these stressors rarely act in isolation, and so they can have cumulative impacts that can often severely threaten wildlife populations up to the scale of biodiversity. Now, in Rockstrom's landmark paper in 2009, in the figure outlined on the left on the screen here, they've outlined 9 planetary boundaries that are critical for regulating the Earth's core ecological processes. And they argued that humans have already surpassed kind of this safe operating boundary, so this green layer in the middle here, for 3 of these major planetary boundaries, one being biodiversity, the second one being climate change, and the third one being biogeochemical cycles like the nitrogen cycle, for instance. So meaning essentially that humans are already having pretty significant impact on several of these core Earth processes. But I'd like to point out here that chemical pollution being one of the 9 essentially hasn't been quantified yet. So they're essentially saying we don't have enough information first to quantify the impacts of this, but not only that, to define the safe operating space with which we can operate. So if we focus first on climate change, we know that climate warming is happening globally, but for the sake of this talk, we're focusing on the Arctic. And what's interesting in the Arctic is that climate change is warming essentially at twice the global average. So in this figure here, the average of the full globe is in brown, and then the green is showing warming in the Arctic. So it's about 2 degrees higher than the global average. And in the figure on the right here, you see kind of the hotspot of the warming anomalies focusing in on the Arctic. So with this temperature increase, which we know is very dramatic in the Arctic, we're seeing severe impacts on almost all components of the physical environment in the Arctic. And one of the most important components that's undergoing this rapid change is sea ice. Now we can see in this figure here, which is looking at sea ice anomalies over time, that essentially over the past decades, we're losing large areas of sea ice in the Arctic because of climate warming. And in extreme years, for instance, that figure on the bottom in 2012, which was a particularly warm year, we can see that the overall extent of sea ice can be dramatically lower than what it was in the past. Now you can imagine this has important implications because sea ice plays such a critical role in Arctic marine and terrestrial ecosystems. Essentially, sea ice is a critical habitat for a lot of ice-associated species in the Arctic, including things like polar bears, narwhal, beluga, and several seal species. Now, not only is sea ice used as an important platform for wildlife foraging and reproduction, it's really also the foundation for a rich ice-associated ecosystem, which starts with ice-associated algae, which feeds fish, which goes all the way up to polar bears, seals, and whales. essentially, when we're losing the sea ice, what we're losing is a critical habitat for these species. And what's also happening at the same time is subarctic species can move northward to essentially take advantage of this new ice-free habitat. And what they're doing is either competing with Arctic species, or they could be acting as important predators. Now, this has been seen for things like killer whales that are moving further north and are important predators for things like beluga and narwhals. Now, the other really particular feature about the Arctic is that it's often referred to as a sink for a lot of global contaminants. Now, this happens because of the fact that many contaminants are produced and emitted in southern, more industrialized regions, but they're transported to the Arctic through major oceanic, atmospheric, and riverine processes. So in this figure on the left here, these major arrows are showing major air masses and how they're essentially moving from the Arctic from everywhere. And the same thing is happening with the ocean, oceanic currents coming from Asia and North America, as well as the North Atlantic, all kind of accumulating into the Arctic. So what that means is that the contaminants that are part of these oceanic or these major transportation processes then get deposited onto the landscape, onto the ice and snow and water, and eventually get taken up by organisms. Now, this is important because things like mercury, but also other persistent compounds, once they enter the marine environment, they can biomagnify to extremely high levels in apex predators. So for those of you who aren't familiar, but the process of biomagnification happens whereby contaminants essentially progressively concentrate at each level of the food web, so that the higher you feed along that food chain, the higher level of contaminants you're going to accumulate in your body. Now, this process is really so important that contaminant levels can reach up to 1 million times or even higher in apex predators like polar bears compared to the level of contaminants in the water. Now, the consequence of this, of course, is leading to really high level of toxic compounds in the bodies of apex predators, like polar bears, whales, and also the indigenous communities that are living in the Arctic and subsisting on these same animals. Now, for mercury, which is really a potent neurotoxin, we know that levels in the brains of many populations in the Arctic are already passing known neurochemical toxicity thresholds. So there's already some level of risk or some level of concern that these species are undergoing neurochemical changes in their brain because of exposure to mercury in the Arctic. So that kind of sums up the main introduction. So now essentially for the remainder of my talk, I'm going to be going through some of the research that I've been working on, again, focusing on these 3 main case studies, with the first one focusing on narwhal. So this first study, it really focused on narwhal in Northwest Greenland, and it's a paper that we recently published in Current Biology. And I was really happy to work on this project because it's such, well, not only is narwhal a really unique and interesting species to work with, but the study design was extremely cool. So here we collected 10 narwhal tusks from hunters in Northwest Greenland. And as you can see in the various pictures here, the narwhal tusk, which is actually just a protruded tooth, it grows incrementally like tree rings, so that when you cut it along its length, like in the figure here, you can see the zoomed in area. What you get is annual growth layer groups. And you can use this essentially to age the animal by just counting up the groups. But what we did was essentially take samples from each of these annual growth layer groups, and we analyzed them for stable isotopes and mercury. Now, stable isotopes are used as dietary proxies, so they tell us a lot about the feeding ecology of the animal. And then, of course, mercury we're interested in because it can tell us something about the exposure and the accumulation of this toxic compound over time. But what was most unique about this study, what I was excited to take part of, especially compared to kind of previous work, is that using these annual growth layers, we could go back in time for each individual animal, essentially giving us a detailed reconstruction of their dietary history and exposure to mercury. And this is something that we just don't have access to just because of the nature of sampling that we have in monitoring programs in the Arctic. And this is essentially what the results look like. So we'll focus first on the stable isotopes data for the 10 individual narwhals, again, going back their entire lifespan. So on the x-axis, we're essentially showing the year at which the isotopes were deposited. So these are the annual growth layer groups. On the y-axis is the isotope values. And then the different colors are different individuals. So for the carbon isotopes, which is the plot on the top, essentially we use this to tell us something about where the animals are feeding. So typically we say that more negative values indicate pelagic or open ocean feeding, and more positive values suggest nearshore or ice-associated feeding. And then for the nitrogen isotopes, higher values typically tell us that these animals are feeding higher up on the food chain. So the important thing to notice here is that it's quite messy. So it tells us something that these animals actually have a high level of dietary plasticity over their lifespan, as well as across individuals. So there's quite a bit of dietary differences between individuals, which is really highlighted on this plot here, showing kind of the dietary niches, where there's quite a bit of overlap in the middle, but you can see that some of these animals are kind of feeding differently from the others. So this is quite nice because previously we had thought that narwhals had a pretty narrow range in their diet, and this is telling us that they can change their diet not only over their lifetime, but individuals in the same population can have quite different diet. Now it's a bit easier to see kind of that general pattern when we just look at the average across all the individuals, and that's what we're showing here. And you'll notice again that the error bars are quite big, meaning there's a lot of inter-individual variability for each year in our data set. But you also notice that the pattern across time isn't really that linear. So the blue line is kind of showing the nonlinear component of the data set, and that's kind of, you can see, quite wavy. So again, their diet is changing over time, kind of in a non-predictable way. So what we're interested in doing is using mixed effect models to try to look at as many different variables as we could to try to explain that variability in the data. And some of the variables that we had included things like the animal age or the calendar year. You can imagine these are correlated, obviously. And then we're also interested in looking at sea ice extent, because we know that the narwhal is an ice-associated species. So we looked at sea ice extent and its habitat. This was Northwest Greenland or Baffin Bay. And then, of course, we had the dietary tracer data. So essentially what we find, and here's the summary of the data is showing the further away from this dotted line, the more important the impact becomes. And essentially what we see is that age or year is a significant factor for both carbon and nitrogen isotopes, meaning that there is a significant temporal pattern in the data. Now for nitrogen, this was positive, and for carbon, it was negative. And then we also found that the isotopes influenced each other. So a change in nitrogen usually comes with a change in carbon isotopes. And this is not too surprising, given that we know that these are dietary proxies, so they likely covary together. But what's more important or more interesting is that we found that sea ice was a significant predictor for both of these dietary tracers and resulted in a positive relationship in both cases. So now if we look at the actual sea ice data, this is what it is for Baffin Bay in the area where the narwhals were sampled. And what we clearly see is essentially around the year 1990 is when sea ice really started to drop consistently. And this essentially aligned pretty well with a drop in both of the other stable isotope data, or the dietary proxies, along that same timeline. So what we're hypothesizing is that a decline in sea ice in the area resulted in a shift in the diet of these animals, where historically they fed primarily on ice-associated and high trophic prey, like Arctic cod and Greenland halibut. And then as the sea ice declines in the area, we're seeing a greater proportion of more pelagic fish, so open ocean fish, specifically things like capelin, which are considered subarctic fish that are moving northward because of climate change. So that was the data on the dietary proxies, but now we'll focus in on the mercury. We did a very similar analysis here and found, again, quite high variability across individuals, but also a very clear pattern with age, where we see that mercury increases progressively as the animal ages. Now, this isn't too surprising for anyone who knows mercury, because it is such a persistent compound. So these animals don't really have the means to eliminate it from their body, meaning that they're going to progressively accumulate it with age. So it was nice to see that, and it was interesting to see that it's a relatively linear pattern when we look at the cumulative age mercury exposure. But of course, we're interested in looking again at which predictor variables were going to explain that data. So we ran mixed effect models for the mercury, and we found again that age and year were very important predictors. So we know that mercury has changes, or essentially has increased over time, and as the animals age. So And also we found that nitrogen isotopes, so the trophic level of the organism, was the other important predictor. So the higher you feed along the food chain, the higher your exposure to mercury will be. So that wasn't too surprising. But what was interesting to us when we looked at the data is this nonlinear pattern where essentially in the most recent decade, we have a more rapid rise in mercury than we had in the historical record. Now you'll remember from the previous slide that during this time period, so the most recent decade, is when the nitrogen isotopes were actually going down, meaning that if diet was driving this change in mercury, we should actually see a reduction in mercury. So this gives us an idea that it's not actually the diet that's causing this rapid increase in mercury in the most recent decade, but likely something else. And essentially what we're hypothesizing is that this most recent rise in mercury is likely the combined effect of increased human global emissions of mercury from things like mining and fossil fuel combustion, but also it could be the effect of climate change, so something a little bit more nuanced, where things like increased release from permafrost, sea ice, and glaciers because of warming in the Arctic, or also increased biological uptake or mercury methylation in the water column in kind of an ice-free, warmer Arctic waters. Now, it's essentially impossible for us to decipher between these two major sources of mercury, but you'll note that both are essentially caused by human activities. So we're pretty confident that some level of human activity is really driving the change in mercury over time. So that summarizes the first study, and now we're going to move on to the second one, in which we looked at beluga teeth. So this was actually quite similar to the narwhal study, but here instead of using incremental growth layer groups along the tusk of the narwhal, we actually look at a full cross-sectional sample of the tooth in the beluga, giving us instead kind of a lifetime average estimate of diet and mercury rather than an annual one. And here our main study site was again in West Greenland on the map here on the top right, where we have samples from hunters along that coast. But what's really unique here for the perspective of this talk is that not only do we have samples from modern times, so from 1985 to 2000, but we have historical samples from museum archives for this area going back to the 1800s. Now this is really cool and really unique as it allows us to look at kind of historical or pre-industrial sources of mercury in these animals. And also you'll notice a few other points on the map. That's because we had data from previous studies on beluga and also ringed seals on Holman Island that we're able to compare to our data set here, and they also had historical samples. So what we end up with is a really kind of unique and strong data set. Now again, I'll first focus on the belugas in Northwest Greenland and on the dietary tracers. So here I'm showing this very similar plots for the carbon and nitrogen isotopes. And you can think of this as a kind of a dietary niche. So this ellipse area is showing kind of a dietary niche for these different time periods, with the blue being historical and kind of the yellowish colors showing more modern times. And what you can clearly see is a pretty nice separation in the dietary niche between the historical beluga population and the more modern ones. And the major difference in this niche is due almost entirely to a change in the carbon isotope signal, which is very consistent with what we saw in the narwhal, meaning that we get a shift from a historically more ice-associated signal to more pelagic or open ocean signal over time. Then we can do kind of some statistics and compare the niche size and the overlap, which is what we're doing in these panels on the right here, to essentially show very similarly that more modern beluga are quite similar to each other in the area and the overlap, and quite different to the historical one. Now, when we combine this data with the data that we had for all the other Arctic populations of belugas and one study of ring seal, which we just threw in there for fun, we get this figure here, where on the extreme right, what we have is the population in Greenland. So you see the shift from the blue to the yellow. And you'll notice that across the Arctic, we essentially have a very consistent pattern where the same shift in carbon isotope signals is happening. So we're getting kind of an increasingly pelagic signal in the diet of these animals, with the exception of the Beaufort Sea beluga population, where we don't really see any directional shift in the dietary tracers. And we believe this is likely confounded by the fact that the Mackenzie River is producing a kind of a huge freshwater input into the area, which is skewing the carbon isotope signal. Now we can also look at mercury in these teeth, which is of course something we're really interested in. And much like the narwhal study, we see that the concentrations of mercury in beluga and ring seal increase with the age of these animals. Again, this is not surprising. We expected this given how mercury accumulates in these animals. However, what we did find is that the rate of increase with age was a lot higher in modern animals than it was in the past. Essentially meaning that mercury accumulates faster in Arctic marine mammals today than they have in the past. So essentially what we can do is correct for this age effect, which is what we did for this plot on the bottom here, which is showing the typical concentration of mercury in a 40-year-old animal across each time period. Again, the blue being historical and the yellow being more modern times. And for this analysis, we can make the assumption that the historical samples represent some kind of pre-industrial baseline, where mass global human emissions of mercury from things like fossil fuel combustion and mining were relatively minimal. So therefore, when we compare to the modern samples, it can give us an estimate of how much of the current mercury is produced from kind of more modern human activities. And what we get is essentially a range from about 80 to 98% of the mercury found in beluga and ring seal today comes from anthropogenic activity. So they're above that pre-industrial baseline. Now, much like the narwhal study, it's impossible to distinguish between those two main sources of mercury. So whether it's direct human emissions from things like fossil fuel combustion, or whether it's kind of more of a nuanced climate change effect where mercury is just changing in the environment and how it's being taken up by the food web. But again, in both cases, we're pretty confident that human activities are to blame for this rather dramatic increase in mercury in marine mammals today. All right, I think we're doing okay with time. So the last study, and this is going to be quite a shift from the previous ones, because now instead of looking at kind of how these things change through time, we're really interested in what the impacts of mercury are going to be on the health of marine mammals. And because mercury, and especially its organic form methylmercury, is so neurotoxic, we focus on the brain for this study. So essentially, we collected whole brains from several marine mammal species, and then we took samples of 10 different brain regions, kind of some of the main ones shown on the slide here. And then for each of these brain regions, we're interested in quantifying the concentrations of mercury, as well as looking at mercury speciation, and then measuring different neurochemical biomarkers like neurotransmitter receptor binding and neurological enzyme function. Now this table is kind of just a quick summary of the 5 species we looked at. So we had polar bears, narwhal, pilot whale, harbor porpoise, and ring seal, and then kind of the sample size. So we had quite a bit from polar bears. These were hunted in East Greenland, and then fewer samples for the other species. So we'll first focus on total mercury across 5 different species. And what we see essentially is a pretty big difference in the concentrations, with polar bears, narwhal, and porpoise having relatively high concentrations compared to polar bears and ring seals. Now this is interesting because you can essentially group these species into 2 major groups. So the first 3 are toothed whales, and then the second ones are kind of fur-bearing species. Now this is interesting because the toothed whales have much higher concentrations than the fur-bearing animals. And this is likely because polar bears and ring seals can get rid of a lot of the mercury by shedding their fur annually. And because we know that fur or hair contains quite a bit of mercury, this is a way that they're eliminating a large source of mercury in their body. And the toothed whales have no such similar mechanism. So essentially, they're retaining their mercury over time. Now this also seems to relate to the species of mercury that we find in the brain of these animals, which is the figure on the bottom, which is looking at the percentage of that total mercury in its organic form, so the percentage of methylmercury. And what we see is essentially the polar bears, and while we didn't have data for ring seal in our study, we have it from previous studies and we know it's quite high, they have a higher percentage of methylmercury compared to the toothed whales. Although you'll notice that the harbor porpoise are a bit of an outlier here, and the percentage of methylmercury is quite high, and we're not really sure why this kind of doesn't fit the pattern as well. However, we do know that methylmercury is the form of mercury that passes that essentially crosses the blood-brain barrier, so it enters the brain. So the differences between species is likely due to demethylation rates in the brain itself. And we don't know a whole lot about species-specific demethylation rates. And then lastly, we can hone in on the different brain regions, which is what we're doing in this figure here, where the brain regions are shown along the x-axis, and again, the total mercury concentration on the y-axis, and then the colors of the different species. And essentially, this is the plot that we get. And the important thing I want you to notice here is that mercury doesn't actually change that much between brain regions, with the exception of high concentrations in the pituitary. And this perhaps isn't too surprising, given that it's the only brain region that we studied that isn't protected by the blood-brain barrier. So a lot more mercury can enter here kind of uninhibited. So now the next step really is looking at those neurochemical biomarkers across all the different brain regions. This is quite a messy plot, and it's kind of summarizing all the data where each panel is showing a different biomarker. The x-axis is showing the different brain regions. The y-axis is showing the change in that neurochemical or the neurochemical levels or value. And then the colors are the different species. Now, I don't intend to go through this kind of panel by panel or anything, but it's simply to show you what we're capable of detecting in terms of brain neurochemistry as it relates to things like receptor binding of important neurotransmitters, as well as enzyme activity, also regulating a lot of these neurotransmitters, and each of these having quite important biological functions. Really, but what we're more interested in is how mercury can potentially affect the changes in these biomarkers. So in a kind of a very rough way, we looked at all the possible correlations between mercury and the biomarkers in all the brain regions for all the species. And what we come up with is essentially a list of 226 possible correlations. And essentially we found 60 significant relationships representing about 27% of the total. And of these, we had some that were positive correlations and others that were negative. So, depending on your perspective here, you could say that we didn't really have a very consistent pattern of mercury-related impacts in these species. However, I selected one of the biomarkers that had the most consistent response and plotted it here, this being NMDA receptor binding. So for this, we typically see that mercury has a negative effect in most species and across most brain regions. Although even here, the results really aren't that consistent. For instance, in harbor porpoises, we see a lot of positive correlations. One thing that we did notice, however, is that the toothed whales who had the greatest concentrations of mercury did tend to have a higher number of correlations than the fur-bearing animals, so the polar bears and the ring seals. So kind of altogether, we can conclude that there's some evidence that mercury could be affecting the neurochemistry in these highly exposed marine mammal populations in the Arctic, but we do see quite a bit of variability across species and brain regions. So we can't really make any conclusive statements about that. So to quickly summarize kind of some of the main points, I'd first say that the teeth in Arctic marine mammals can provide us a very useful and unique archive of ecological data that we can use to ask really kind of important and interesting questions about climate change impacts on contaminants over time. What we found is that climate change impacts on contaminant trends, what we found is something very consistent across a lot of other species in regions in the Arctic, which is an increasing proportion of subarctic or pelagic prey in these species with climate change, and essentially with the loss of sea ice. We've kind of indicated that a change in diet can have important implications for contaminant accumulation. But more importantly, it seems that the major changes, especially in mercury, are due to human activities and a little bit less due to diet changes, especially when we compare it to historical samples going back to the 1800s. And of course, the largest unknown here really remains to be, do these changes in diet and exposure to mercury have any meaningful impacts on these species? And this is really an open-ended question. We essentially need a lot more work to tackle this question and especially consider the cumulative impact of multiple stressors, because we know that these animals are being stressed by a lot of other factors. So with that, that's the end of my talk. I'll just finish by acknowledging all the great people that made these projects possible, including the hunters and the Arctic communities in Canada and Greenland, as well as all my collaborators in Canada, in Europe, and of course, all the different funders that have supported this work. So with that, I'll just thank everyone for tuning in, and please don't hesitate to ask questions or get in touch with me if you have any questions.
Thank you, Jean-Pierre, for your very interesting presentation. I encourage people to either put up their hand or to put their questions in the Q&A or in the chat. Je vous invite de poser des questions soit en anglais ou en français. Tu peux lever la main ou mettre vos commentaires dans la boîte de clavartage. I think I'll start. I know very little about marine mammals, so I was just wondering if there's ever been a comparison between southern belugas and northern belugas in terms of you know, accumulation of contaminants, or are they subspecies of the same thing, of beluga?
Yeah, that's a great question, and it kind of highlights one of the most interesting group of studies that really got me interested in marine mammals to begin with. Was some of the work by Sylvain Deguise and colleagues in Quebec that were comparing exactly this. So the population of belugas in the St. Lawrence to the populations in the Arctic, kind of using it as a kind of a case control study where you can think of the Arctic as a pristine area where there's relatively few human activities that can stress them out, and then the St. Lawrence being the complete opposite. So we know that that beluga population has been endangered for quite some time and has been stressed by multiple things, including a lot of contaminants. And a lot of those studies were focused on things like tumors and the immune system. And essentially they found a very consistent pattern where the beluga populations in the Arctic, which are relatively pristine and have few contaminants in their bodies, are a lot healthier than the ones in the south. And kind of since then, very similar studies have been done across the world in other species. So my colleagues in Germany, for instance, look at porpoises in the North Sea and compare that to those around Greenland and find the same thing. There's a much higher incidence of disease, suppressed immune system, and a lot more parasites and things like that in the south than there are in the north. And these kind of case studies are exactly the studies that we need to ask these kinds of questions, because just focusing on one population can't really get you that same kind of information.
And contaminants like mercury, for instance, do they affect the lifespan of an animal? Has, you know, first of all, what is the lifespan of a narwhal or of a beluga under, as you say, pristine or optimum conditions? And you know, for instance, does mercury, because it is a neurotoxin, shorten that lifespan? Or it's hard to tell probably because there's so many things that interfere.
Yeah, I mean, your last point is exactly right. I mean, I wish I had a better answer for you. We do know something about the lifespan of these animals. And for instance, the largest tusk we found in our study had over 50 annual layers. So we know that the animal lived at least 50 years, but part of the tusk was broken, so it's likely even older than that. So these are long-lived animals. But as you mentioned, there's so many factors that come into play, and just the fact of how we sample these animals, we simply don't have the information to look at whether these contaminants are affecting the lifespan. Kind of the best we can do is look at things like, you know, can these affect reproduction, or can they suppress your immune system, or look at more molecular level impacts. It's very hard to get to these really larger questions like, you know, population demographics and survival.
Okay, going to the feeding plasticity, do we know if narwhals or belugas will search out the benthic and the ice, you know, the ice edge species? Do they migrate a little bit? Because I would imagine that is their preferred food is ice species. do we have data as to whether they migrate to look for those species, or do they sort of, are populations distinct and sort of stay in one region?
Yeah, so especially with the narwhal, they definitely migrate seasonally. So they have kind of a winter habitat and a summer habitat. And in the summer, they migrate towards the kind of Canadian archipelago into narrow channels to feed for these fish. So, you know, it's a lot of factors that come into play. I mean, areas they like for pupping and you know, also it's avoiding predators. So they're trying to get areas where the killer whales aren't following them. So a lot of these things kind of interact in terms of habitat selection. But we know from tracking studies, so people are putting satellite tags on these animals, and we're able to identify the habitat that they like, which is typically in a certain temperature range, which seems to be an important factor for narwhal. So as climate changes, it's very likely that these animals are going to track their optimal temperature range that they're looking for. And that remains to be the big question is are their habitat choices going to change with climate change, or are they going to be very kind of stuck in the previous ways? But satellite tagging efforts have shown that, you know, they do move across large areas. So there's nothing to suggest that they're stuck in one particular region. And the bigger question is how selective are they? And the data seems to suggest that they can change their prey as the prey base changes. So, you know, as capelin and other fish kind of move northward, they seem to be feeding on them more. So, you know, very likely there's some level of generalist feeding, so they'll eat whatever's available to them. And the remaining question becomes, are the kind of subarctic species moving northward as good for them as the Arctic species? So, you know, are they getting enough energy from feeding on these fish compared to kind of the lipid-rich fish that live in the Arctic? And that remains to be kind of an open question.
Well, thank you very much. Do we have any questions for Jean-Pierre? Est-ce qu'on a des questions pour Jean-Pierre, soit en anglais ou en français? No, but you have a large fan club that is attending today. I just want to thank you again for your presentation. And will you be continuing this type of research when you move to the University of Winnipeg?
I certainly hope to. So, I mean, we have a lot of open questions, and you know, there's different isotopes that we can look at, and you know, looking at mercury in more detail throughout the narwhal tusk, and just using teeth in general as kind of a unique temporal archive of data is something that I'd like to continue with. But especially looking at the impacts of chemical stress is something that I'm really going to focus on in my new position.
Well, we wish you the best of luck at the University of Winnipeg, and let us know if you're looking for grad students. I'm sure that we can put the message out. And okay, thank you again, Jean-Pierre. And I invite everyone to check out the ENQ website for our upcoming webinars, and they will also be listed in the ENQ infolette. So see you all for our next webinar. Thank you again, Jean-Pierre. Bye, everybody.